Usually when we talk about a medical diagnosis, there's this expectation of mechanical precision, like engineering.
Right, yeah, like fixing a car.
Exactly. You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is, broken.
It's a comforting binary, you
Yeah.
know. We like things to be visible, we like them to be categorized, and most importantly, to be fixable in a really straightforward way.
Yeah.
A bone breaks, you set it, it knits back together.
But then you step into the world of human biology, specifically the internal organs, and suddenly that x-ray machine feels, well, completely inadequate.
Oh, absolutely.
We're looking at a landscape that is, honestly, it's terrifying when you realize the limitations of our own bodies. Let's talk about the human kidney today.
A fascinating organ.
It really is.
Yeah.
It is an incredibly complex filtration plant, just an absolute marvel of biology, but it has a fatal flaw.
It does.
Its regenerative capacity has a hard biological stop.
A remarkably sudden stop, biologically speaking. I mean, by the 36th week of gestation.
Wait, 36th week?
Yeah, before you were even born, human nephrogenesis just concludes.
Which means no new nephrons are ever formed for the rest of your life.
Exactly.
You are born with all the filtration units you will ever have. It's an endowment.
Right, and it varies, you know. Some people might be born with 2 million, some with maybe 500,000, especially if they were born premature.
But whatever your number is, that's it.
That is your limit.
If they are damaged by disease or trauma or, I don't know, a toxic medication, they are gone. There is no biological backup plan, right?
Yeah.
No stem cells waiting in the wings to build you a new filter.
None. And the gravity of that limitation is staggering when you look at the global health landscape.
Yeah, the numbers are rough.
We are dealing with a burden where approximately 10% of the entire world's population suffers from chronic kidney disease.
10%. I mean, that's almost 800 million people walking around with a deteriorating filtration system.
And it frequently progresses to end-stage renal disease.
Which is when the kidneys just give out.
Right. At that point, the natural filtration system has fundamentally failed. The body begins essentially poisoning itself with its own metabolic waste.
Ooh, that's a brutal reality.
It is. And the only current fixes are incredibly costly, invasive dialysis or, waiting for a rare organ transplant.
Because there just aren't enough organs to go around.
Exactly. We have a massive, seemingly insurmountable mismatch between the supply of donor organs and the growing population of patients in need. And dialysis, while it's life-saving, it's brutal.
Yeah, it's not a cure.
No, it only replicates a fraction of a healthy kidney's function.
So to solve this, scientists are pulling off what looks like a biological miracle.
It really does feel like magic sometimes.
Today on The Deep Dive, our mission is exploring this massive stack of recent scientific literature about a transformative shift in regenerative medicine.
And it's a huge shift.
We're going to explore the generation of human, pluripotent, stem cell-derived kidney organoids. I mean, we are literally talking about taking human cells, hitting reverse, and growing functional kidneys in petri dishes.
What's fascinating here is that researchers are no longer relying solely on flat, two-dimensional cell cultures.
Right.
Or animal models like mice that, consistently fail to accurately mimic human biological responses.
Because mice aren't humans.
Exactly. They are now coaxing stem cells to self-organize into three-dimensional, miniaturized, functional kidney structures. It is a complete paradigm shift.
And I want to promise you, the listener, right now, whether you are a medical professional looking for the latest on nephrotoxicity screening, or just someone curious about the future of human longevity.
Because it affects all of us.
Exactly. Understanding how we are learning to regrow our most complex organs will fundamentally change how you view your own biology.
It really will.
Okay, let's unpack this, starting with the biological blueprint of the human filter.
Good place to start.
Because to understand why building a replacement is so hard, we have to look at how insanely complicated the original equipment is.
Right. The human kidney isn't just a sack of uniform cells acting like a coffee filter.
A coffee filter is pretty simple.
Yeah, when water passes through a paper filter, it's a passive process based purely on size. But the kidney is an active, highly intelligent sorting facility.
How many different cell types are we talking about?
It comprises more than 20 distinct cell types.
Wow. And they are all intricately organized into functional units known as nephrons.
Yes, the nephrons do the heavy lifting.
Handling metabolic waste filtration, sure. But they are also managing electrolyte homeostasis, like balancing your sodium and potassium.
Right, and they even have endocrine functions.
Like releasing hormones.
Exactly. They release hormones to regulate your blood pressure, and they even tell your bone marrow to make more red blood cells.
So they are the ultimate multitaskers of the human body.
Oh, absolutely.
I was reading through the source material, and I kept picturing the kidney as this highly secure, multi-stage water treatment facility.
I like that analogy.
It's not just one big sieve, right?
Oh.
You have different stations handling very specific molecular sorting.
That's a highly accurate way to visualize it. The fluid dynamics are just incredible.
Right. So first, the blood comes into the glomerulus.
The first station.
Yeah, I picture this as a knot of microscopic hoses. And wrapped around these hoses are these specialized cells called podocytes.
Podocytes, yes.
They act like the primary grate. They physically interlock their little, I think they're called foot processes.
Yes, they're foot processes.
Yeah.
They interlock to keep large vital proteins inside the bloodstream while letting water and toxins pass through.
And right after that primary grate, the filtrate travels through the proximal tubules.
The next station.
Right. If the glomerulus is the grate, the proximal tubule is like the recycling sorter.
Exactly. It uses active transport to grab back the good stuff.
Like what?
Like glucose, amino acids, bicarbonate. It pulls them out of the waste stream and puts them back into the blood.
So smart. Then the fluid dives down into the loop of Henle, which is all about water concentration.
Pumping salts to create a gradient.
Right. It sucks water back into the body so we don't dehydrate.
Crucial for land animals, yeah.
And finally, the distal tubules do the fine-tuning of the electrolytes before the remaining fluid becomes urine.
And the thing is, if any one of those stations goes offline...
The whole facility is compromised.
Exactly. Because this system is so uniquely specialized and so dependent on the spatial relationship between these different cell types, traditional research methods have, well, they've consistently failed us.
Right, because for decades, the gold standard in renal research was the murine model, mice.
Yes. But as we discussed...
Mice are not tiny humans. We've known that for a long time.
We have, but the extent of the divergence is what's shocking.
How divergent are we talking?
When we look at the transcriptomic profiles, the exact gene expression signatures, during acute inflammatory responses or, say, a drug-induced injury, murine models wildly diverge from human responses.
They just react totally differently.
Right. The metabolic pathways in a mouse kidney have evolved for a creature with a completely different lifespan, a different diet, and a much faster heart rate.
So a drug might work perfectly in a mouse.
A drug that perfectly clears a toxin in a mouse might completely destroy the proximal tubules in a human.
Wow. Which perfectly explains why a pharmaceutical company can spend a decade and $1 billion developing a kidney drug that looks amazing in mice.
Only to watch it fail spectacularly in phase 2 human clinical trials. It happens all the time.
But wait, if we've known how important the 3D environment is, why hasn't science moved past mice and petri dishes sooner? I mean, what about just growing human kidney cells in a dish?
I see the HK2 cell line mentioned a lot in older literature.
Researchers certainly tried. They isolated human kidney cells, specifically those proximal tubule cells, and grew them on flat plastic dishes.
But those are 2D cultures.
Exactly. And the literature definitively shows that two-dimensional cultures completely lack the three-dimensional biomechanical and biochemical microenvironments needed for cells to behave naturally.
They lose their context.
Exactly that. Imagine taking a brick out of a vaulted ceiling and laying it flat on the ground.
It's still the same brick, but it's no longer performing its function because it lacks the surrounding pressure and spatial geometry.
That makes perfect sense.
When you take a highly specialized tubular cell and stick it flat on a piece of hard plastic, it loses its normal polarity.
Polarity, like a magnet.
Sort of, but spatial. In the body, a tubule cell has a top that faces the urine and a bottom that faces the blood.
Oh, right.
On flat plastic, it gets confused. It stops expressing the critical transporter proteins it uses in the body. It essentially forgets how to be a kidney cell.
That is wild. So mice have the wrong genetics, and 2D human cells have the wrong geometry.
You need both the right genetics and the right geometry.
It seems obvious in hindsight that we needed 3D human tissue. What took so long? What was the missing biological key?
The missing key was understanding the exact step-by-step embryological recipe to direct stem cells. We didn't know the instructions.
So we had to reverse engineer nature.
We had to figure out how nature builds a kidney from scratch in the womb, decode the chemical signals used in that environment, and then meticulously recreate those signals in a lab setting.
The developmental cookbook. I love that concept. Let's dive into that kitchen.
Okay, let's do it.
To build these organoids, the sources show scientists use human pluripotent stem cells,
Yes.
HPSCs.
Yes.
These can be embryonic stem cells or
Yeah.
incredibly induced pluripotent stem cells, iPSCs.
The advent of iPSCs is arguably one of the greatest biological triumphs of our century.
It sounds like science fiction.
It really does.
Yeah.
These are adult cells, say, a simple swab of your skin cells or a routine blood draw that have been genetically reprogrammed.
Just normal skin cells.
Yeah. By introducing a specific cocktail of transcription factors, scientists wipe the cell's memory. They reverse its epigenetic clock, turning it back into an embryonic stem-like state.
It becomes a blank slate again.
A completely blank slate.
So you have this blank slate sitting in a dish. How do you turn it into a kidney? The literature cites breakthrough protocols developed by researchers like Takasato, Morizine, and Taguchi.
Yes, the pioneers of this method.
They call it directed differentiation.
Directed differentiation is the art of walking these amnesiac stem cells down the exact developmental pathway they would naturally take in a developing embryo.
So it's very controlled.
Highly controlled. It relies on the sequential, highly timed application of specific biochemical signals. It is highly choreographed.
It's like dance.
Exactly. If you add a chemical a day too early or wash it away a day too late, you don't get a kidney.
Walk us through the recipe. What is the first step?
The very first step is to apply a biochemical signal to the pluripotent stem cells, specifically a small molecule called CHR99021.
CHR99021.
Sounds like a droid from Star Wars.
It does, doesn't it?
What does it actually do to the cell?
It's a targeted inhibitor. In biology, you often activate a pathway by blocking the thing that normally stops it.
Like taking the brakes off a car or silencing a security guard. CHIR99021 inhibits an enzyme called GSK3-beta.
Okay.
By blocking this enzyme, it activates an ancient evolutionary communication highway known as canonical Wnt signaling.
One signaling. That pops up everywhere in developmental biology.
Because it is fundamental to how multicellular organisms determine their body plan.
Right.
By exposing the stem cells to this specific chemical bath for a precise number of days, you push them to become what's called the primitive streak.
The primitive streak.
In an embryo, the primitive streak is the very first branching point where a ball of identical cells starts organizing into distinct layers that will eventually become, the nervous system, the gut, or the organs.
Here's where it gets really interesting to me. It's like biochemical origami.
That's a great way to put it.
You don't just physically fold the paper with tiny tweezers.
Yeah.
You expose the paper to specific chemical baths at the exact right moment. And the cells literally know how to fold themselves.
That inherent knowledge that self-organization is the defining hallmark of an organoid.
We aren't building it.
No, we aren't building the kidney cell by cell. We are providing the cues, and the cells execute their ancient programming.
Incredible. So after the primitive streak,
Once they reach that primitive streak stage, you have to change the chemical bath. You introduce growth factors like FGF9, fibroblast growth factor 9, and often another protein called BMP7.
and what are FGF9 and BMP7 telling the cells to do?
They are acting as molecular signposts, guiding the cells down the mesodermal lineage.
Telling them what not to be.
Exactly. They tell the cells, you aren't going to be a brain cell, and you aren't going to be a lung cell. You are going to be intermediate mesoderm.
Okay, narrowing it down.
And from there, with continued chemical coaxing, they become metanephric mesenchyme, or MM.
Ananephric mesenchyme. I saw that term heavily emphasized in the sources. That's the crucial progenitor tissue, right?
Yes.
It's the raw biological clay from which the nephrons are going to be molded.
It is the absolute prerequisite. Once you have successfully generated a high percentage of these metanephric mesenchyme cells, the physical environment changes.
Oh.
Researchers often transfer them from a flat culture surface into a specialized 3D plate or a suspension culture where they float.
So they aren't stuck to the bottom anymore.
Right. This forces the cells to aggregate, to clump together into a tight spheroid, and then a spectacular biological transition occurs. The cells undergo a process called mesenchymal to epithelial transition, or MET.
Mesenchymal to epithelial transition. Let me try to visualize this. Mesenchymal cells are sort of loose, mobile, individual cells,
You're
right?
mobile.
Like a crowd of people milling around a plaza. But But epithelial cells are structured. They form tight barriers and tubes.
Exactly.
So MEST is like that loose crowd suddenly deciding to lock arms and form a highly organized, impenetrable wall.
That is a perfect visualization. Through MET, the loose progenitor cells lock together and self-organize into defined structures,
Without anyone touching them.
without any physical scaffolding built by human hands. The cells communicate via chemical gradients. They start forming segmented nephron structures.
That is so fast.
Within weeks, you can look under a microscope and see actual glomerular podocytes forming that knot, feeding into a proximal tubule, extending into a loop of Henle, and finishing in a distal tubule.
All in their correct spatial orientation.
All correctly oriented.
I want the listener to really think about this for a second. Imagine your own skin cells.
Just a scrape from your arm.
Right. Imagine a simple skin biopsy being reverse engineered back to a microscopic blank slate. Then, those cells are chemically coaxed day by day over about 3 to 4 weeks to grow, differentiate, and literally fold themselves into a tiny living version of your own kidney sitting in a lab dish.
It represents a profound leap in our mastery of cellular biology. But, generating these organoids is not just an academic exercise in proving we can play the notes of developmental biology.
No, the real-world applications are immediate.
Yes, they are critical.
Right, because while growing a healthy mini kidney is a cool party trick, a monumental achievement, the real earth-shattering value is what happens when you grow a sick kidney. A healthy organoid only tells us how things work when they go right. To actually cure patients, scientists had to figure out how to intentionally break these organoids to mimic genetic diseases.
And that leads us directly into the realm of disease modeling, which is perhaps the most immediate triumph of this technology.
Because animal models were failing us here too.
Oh, massively. For a long time, studying genetic kidney disorders was hampered because, as we discussed, animal models didn't perfectly replicate the human phenotype.
So a mouse with a genetic mutation might not actually get sick.
Or they might not develop the same symptoms as a human with that exact same mutation. It made drug testing incredibly frustrating.
The sources focus heavily on one disease in particular to prove this point: autosomal dominant polycystic kidney disease, or ADPKD.
Yes.
It's the most common hereditary kidney disease worldwide.
It accounts for up to 10% of all end-stage renal disease cases globally.
10%.
And for those unfamiliar with the pathology, ADPKD is a devastating condition.
It sounds awful.
It is characterized by the relentless growth of massive, fluid-filled cysts within the kidneys. A normal human kidney is about the size of a human fist.
Okay.
An ADPKD kidney can grow to the size of a football and weigh up to 30 pounds.
30 pounds?
Yes, entirely consumed by these cysts. It crushes the healthy tissue, inevitably requiring the patient to undergo dialysis or transplantation.
And the tragic part is that it's hereditary. It's written into the patient's DNA.
Exactly.
Specifically, it's caused by mutations in the PKD1 or PKD2 genes.
To understand how a tiny genetic typo leads to a 30-pound cystic kidney, we have to zoom way in on the cellular mechanics.
Let's zoom in.
If we look closely at the tubular epithelial cells, the ones lining the inside of those tubules we discussed earlier, they have these tiny, solitary, hair-like projections extending into the hollow space of the tube.
Like little hairs.
Right. These are called primary cilia.
I think of them as cellular antennas, waving around in the urine stream, sensing fluid flow and picking up chemical signals.
That's functionally what they are. Now, the PKD1 and PKD2 genes encode two very specific proteins, polycystin 1 and polycystin 2.
And where do those proteins go?
These two proteins link together to form a complex that sits right in the membrane of that primary cilium antenna. And this complex operates as a calcium-permeable channel.
So it's a sensory gatekeeper for calcium.
Yes. Here is the mechanism of how it's supposed to work. As fluid flows through the kidney tubule, it physically pushes against the cilium.
The antenna bends.
Exactly, the antenna bends. That mechanical bending forces the polycystin channel to pop open, allowing calcium ions from the fluid to rush into the cell.
Okay, so calcium enters the cell. What does that do?
In this specific context, that influx of calcium acts as a molecular chill-out signal.
Chill out.
Yeah, it travels to the nucleus and tells the cell to maintain its normal flat structure and, most importantly, not to multiply.
Stay calm, stay flat, don't divide.
Exactly. But when a patient has a mutation in PKB1 or PKD2, those polycystin proteins are malformed.
The calcium channels are busted.
The antenna might still bend, but the gate doesn't open. Calcium doesn't enter the cell.
Oh.
And when the cell is deprived of that calcium signal, it misinterprets the silence. It triggers a cascade of panic signals inside the cell.
Because it thinks something is wrong since it's not getting the signal.
Right. Specifically, the lack of calcium hyperactivates an enzyme called adenyl cyclase, which leads to a massive spike in a molecule called intracellular cyclic AMP, or CAN-AMP.
And what does a massive spike in CAMP do to the cell physically?
It does two catastrophic things simultaneously. First, it turns on protein kinase A, which drives excessive cell proliferation.
So the cells start dividing rapidly.
Yes, piling up on each other instead of staying in a neat single layer. Second, it stimulates CFTR chloride channels to start aggressively pumping chloride, and water always follows chloride into the center of this mass of dividing cells.
Wait, so you have a ballooning cluster of rapidly dividing cells, and at the same time, they are furiously pumping fluid into a closed space.
Yes.
The balloon inflates. That is exactly how a cyst forms.
That is the complete mechanotransduction pathway from a genetic mutation to a physical cyst. Now, for decades, trying to find a drug to stop that CAMP cascade was agonizingly slow.
Because of the mouse models.
Exactly. But organoids changed the game. Researchers brought in the heavy artillery of modern biology. CRISPR-Cas9 gene editing.
Oh, CRISPR. That's like highly programmable molecular scissors. You can cut DNA at a very specific location.
And using CRISPR, researchers can do two incredible things with these stem cells. They can take a line of perfectly healthy stem cells and use CRISPR to intentionally break the PKD1 or PKD2 genes.
Just break them on purpose.
Right, introducing the exact mutation found in patients. Or conversely, they can take iPSCs derived from an actual ADPKD patient, someone who already has the disease, and use CRISPR to fix the mutation.
You fix the typo in the lab.
Yes, that creates what the literature calls an isogenic control. It's a perfectly matched, healthy version of that specific patient's own tissue, differing by only that single genetic correction.
That's brilliant. So they take these stem cells with the broken antennas and they run them through that developmental cookbook we talked about. They grow the organoids.
And when they do, the results are visually startling.
What happened?
Under the right conditions in the lab, often by adding a chemical like forskolin to artificially increase that CAMP panic signal, these tiny microscopic kidneys literally form massive fluid-filled cysts right there in the petri dish.
Wow.
It is a perfect human-specific disease-in-a-dish model. It mimics the human pathology flawlessly.
And once you have the disease thriving in a dish, you can attack it.
Exactly.
And because these organoids are small and can be grown from essentially limitless stem cells, you don't just have one sick kidney to study, you can generate thousands of them.
Yes, scalability is key.
The sources highlight incredible high-throughput screening successes. We're talking about growing these mutant organoids in 96-well or even 384-well plastic plates.
A microscopic cystic kidney in every single well.
And because they are in these standardized multi-well plates, scientists can turn the testing over to automated robotic fluid handling systems.
The robots take over.
Right. A robot can systematically drop a different experimental pharmaceutical compound into every single well, simultaneously testing hundreds or thousands of drugs to see if any of them shrink the cysts or stop them from forming in the first place.
The efficiency is unmatched.
The literature mentions a specific study by Tran et al., where they generated these organoids embedded in methylcellulose to hold their shape during high-throughput screening.
Yes, a very important study.
They ran a vast library of compounds and identified several known cyst inhibitors, which validated the model. But they also found a novel compound called quinazoline that actively reversed the cyst growth.
Reversed it.
Yeah. Another study used a small molecule library and found that metformin, which is a very cheap, very common diabetes drug that millions of people already take safely, showed a dose-dependent inhibition of cyst formation in the organoids.
If we connect this to the bigger picture, the implications for pharmaceutical development are staggering.
How so?
Well, traditionally, to test those hundreds of compounds, you would need thousands of mutant mice. You would have to feed them the drugs for months or years, then sacrifice them, dissect the kidneys, and manually measure the cysts under a microscope.
That sounds incredibly slow.
It costs millions of dollars and decades of time. And even then, the mouse biology might yield a false positive. With CRISPR-edited organoids, we skip the ethical dilemma and the biological hurdles of animal testing. We are fast-tracking personalized precision medicine.
It's mind-blowing to think about the timeline. I mean, if a patient is diagnosed with a specific genetic variant of ADPKD,
Right.
you could hypothetically take their blood, make iPSCs, grow organoids, test 50 different targeted drugs on their own tissue in a matter of weeks, and find the exact molecule that works for their specific mutation.
That is the ultimate goal of precision nephrology, not just treating the disease, but treating the disease as it exists in your specific genetic landscape.
But genetics are only one piece of the puzzle, right? A healthy kidney can be destroyed overnight by something completely outside your DNA.
Unfortunately, yes.
In fact, a huge danger comes from the very places trying to heal us. I was looking at the stats on hospital-acquired acute kidney injury, and the numbers are frightening.
It's a silent epidemic.
So we have this perfect testing platform. Let's talk about the front lines of toxicology, nephrotoxicity, and drug screening.
This is a massive, often under-discussed issue in clinical medicine. The kidneys are exceptionally vulnerable to drug-induced kidney injury, or DKI.
DKI, why are they so vulnerable?
You have to consider the sheer volume of blood they process. The kidneys filter about a quarter of your entire body's blood volume every single minute.
That's a huge volume.
And their job is to concentrate toxins in order to excrete them in the urine. Because of that concentration process, the epithelial cells in the kidney are exposed to incredibly high, sometimes toxic levels of circulating pharmaceutical drugs.
So they take the brunt of it.
Exactly. DE is a leading cause of acute kidney injury in hospitalized patients who are receiving heavy courses of antibiotics or chemotherapy.
And discovering that a new miracle drug is actually nephrotoxic during a late-stage human clinical trial, that is an absolute nightmare for pharmaceutical companies.
A multi-billion-dollar nightmare.
Right, you've spent billions of dollars getting the drug out of the lab, it cured the mice, and now it's causing kidney failure in human patients. They desperately need a way to test drug toxicity early on.
They do.
But we mentioned earlier that traditional 2D flat cell lines, like the HK2 line, fail at this. We talked about them losing their polarity, but how does that actually prevent them from detecting a toxic drug?
To understand why 2D cells fail to detect toxicity and why organoids succeed, we have to look at the physical mechanics of how a drug actually gets into a kidney cell.
Okay, let's look at that.
Drugs don't just passively float like ghosts through the cellular membrane. There are complex molecules, often carrying an electrical charge.
So they need a door.
Exactly. They have to be actively pulled inside the cell by highly specialized transporter proteins. In the kidney's proximal tubules, the key architectural doorways for this are the SLC22 transporter family.
The sources specifically highlight OAT1, OAT3, and OCT2.
Yes. Organic anion transporters, the OATs, and organication transporters, the OCTs. These are the literal molecular revolving doors.
Where do they sit?
They sit on the blood-facing side of the tubule cell. An OAT will grab a molecule of alpha-ketoglutarate from inside the cell, push it out into the blood, and in exchange grab a drug molecule from the blood and pull it inside.
Chemical swab.
Right. The cell then shuttles the drug across its interior and pumps it out the other side into the urine.
So if a drug is going to poison the cell, it first has to use these doorways to get inside.
Precisely. Now, when you take those human tubular cells and culture them flat on a piece of plastic, the cells undergo a stress response.
Because they're in the wrong environment.
Yes. Because they lack the 3D architecture, they drastically downregulate the expression of genes that build OAT1, OAT3, and OCT2. They dismantle the revolving doors.
If the cell doesn't have the doorway, the drug just bounces off the exterior. It can't enter the cell's cytoplasm.
Exactly.
And if the toxic drug can't enter, the cell doesn't die. So a pharmaceutical scientist looking at that 2D Petri dish sees happy, healthy cells and assumes the drug is perfectly safe.
It's a massive false negative. The drug is highly toxic, but the test system is completely blind to it.
That is terrifying.
But organoids are fundamentally different. Because organoids develop in a 3D environment that mimics embryonic development, with all the correct spatial cues, they properly express these SLC22 transporters.
The doors are there.
The doorways are fully built and wide open. Therefore, organoids act as highly sensitive, biologically accurate alarm systems for drug toxicity.
The sources provide some very stark real-world examples of this biological alarm system in action. They tested cisplatin on these organoids.
Yes, cisplatin.
Which is a very potent, widely used chemotherapy drug for solid tumors. But it is notoriously brutal on the kidneys of cancer patients.
It is a perfect test case for this technology. When researchers applied cisplatin to the fluid surrounding the human kidney organoids, the organoids reacted exactly like a human patient.
How so?
The cisplatin was pulled into the cells via the OCT2 transporters. The organoids showed dose and time-dependent damage, specifically targeting the proximal tubule cells.
So they died.
Yes, the cells underwent apoptosis-programmed cell death, and they began expressing specific biomarkers of acute kidney injury, like hemodynamine.
They also tested tenofovir, an antiviral medication used for HIV and hepatitis B, and aristolochic acid.
Aristolochic acid is fascinating.
Yeah, it's a highly potent nephrotoxin found in certain plant species that were traditionally used in herbal medicines. It ended up causing a global wave of kidney failures before it was identified.
Or a real tragedy.
And in every single test case, the organoids successfully imported the toxin and flagged the toxicity, dying exactly the way native human tissue would.
The paradigm shift here is economic as well as medical.
How so?
Moving forward, pharmaceutical companies can integrate kidney organoids as a mandatory screening step in preclinical development. Before a newly synthesized drug is ever given to a mouse, let alone a human in a clinical trial, it can be flushed through arrays of human kidney organoids.
And if they died?
If the organoids show early markers of DNA damage or apoptosis, the chemists can modify the drug's molecular structure to make it less toxic or scrap it entirely.
Saving billions of dollars.
Billions of dollars and preventing untold human suffering.
Okay, I have a major question here, though.
Go for it.
An organoid sitting in a multi-well plate is just sitting there. It's immersed in a static pool of nutrient broth.
Right.
It doesn't have a beating heart pumping pressurized blood into its glomerulus. It doesn't have a bladder to drain its filtrate into. The fluid dynamics are zero. How can we possibly test drug toxicity accurately if there's no blood flow to deliver the drug dynamically and no urine flow to wash away the cellular waste? Doesn't the lack of plumbing compromise the biology?
That is an excellent point, and that is the exact roadblock the scientific community hit once the initial excitement wore off.
Really?
Yes, you have identified the primary limitation of static culture. Culturing organoids in a static dish of fluid results in what the literature calls fetal-like or immature organoids.
Fetal-like, meaning they don't grow up.
Exactly. They develop the right structures, the tubules and the podocytes, but developmentally they stall out at a stage resembling a first trimester embryonic kidney.
So they're kidneys, but baby kidneys.
Right. Their gene expression isn't fully mature. To solve this, developmental biology had to reach across the aisle and team up with mechanical engineering.
Which brings us to the bioengineering rescue. We have to build the plumbing ourselves.
We do.
The literature focuses heavily on two massive advancements: organ-on-a-chip technology and 3D bioprinting.
To get these fetal organoids to truly mature into adult-like tissue, we have to subject them to the invisible physical forces they would experience inside a living, breathing, pumping body.
Because the body isn't static.
No, the body is highly dynamic. The current limitations of static organoids include that incomplete maturation, a lack of vascularization, meaning no blood vessels are penetrating the tissue to feed it, and the absence of a fluid drainage system. The most elegant solution to this is microfluidics.
Organ-on-a-chip. I mean, it sounds like something out of a cyberpunk novel. What does this chip actually look like?
It's not a silicon computer chip. An organ on a chip is a microfluidic device, usually made of a clear, flexible, biocompatible polymer called PDMS.
How big is it?
It's small, about the size of a USB thumb drive, and it contains tiny, engineered, hollow microchannels. Researchers take a developing kidney organoid, this tiny sphere of tissue, and carefully place it inside one of these chambers.
Oh, no way.
Then they hook the chip up to micropumps, and these pumps constantly, rhythmically flow nutrient-rich liquid media past the organoid.
So you are mechanically simulating the pulse and flow of blood.
Yes. And this introduces a critical physical force that is entirely absent in a petri dish. It's called fluid shear stress, or FSS.
Fluid shear stress. That's the friction of the liquid dragging against the surface of the cells as it moves past them.
Exactly. And the cells can actually feel it.
Wait, feel it.
Remember those primary cilia we talked about earlier? The antennas.
Oh, right. The antennas that bent to let calcium in.
Yes. When the micropumps turn on, the fluid flows, the antennas bend, and mechanotransduction occurs. The physical force is translated into chemical signals. Studies from pioneering researchers like Hallman and Morzane demonstrate that subjecting organoids to fluid shear stress dramatically alters their genetic destiny.
It forces them to mature.
It does.
How so? What physically changes inside the organoid when you turn on the flow?
The most profound and necessary change is vascularization.
Blood vessels.
Yes. A static organoid has endothelial progenitor cells, the cells that want to become blood vessels, but without flow, they just sit there dormantly.
Waiting for a signal.
Exactly. Under dynamic fluid flow, The sheer stress triggers the release of vascular endothelial growth factor, or VEGF. The endothelial cells wake up. They start multiplying and form complex branching networks of capillaries.
They start building their own blood supply.
They do. And even more incredibly, under flow, these newly formed blood vessels begin to actively invade the podocyte clusters in the glomerulus.
Wait, they hook themselves up to the filter.
It's a beautiful biological synergy. They wrap around the tubular segments, improving cellular polarity, giving the cells a clear blood side and urine side, and boosting the formation of cilia. The physical friction of the fluid pushes the organoid out of its fetal stalling point and forces it to structurally and genetically mimic an adult tissue.
That is just incredible, but the microfluidic chip is only one part of the bioengineering rescue.
It is.
Because if you want to test thousands of drugs, you need thousands of chips, and you need the organoids inside them to be identical. I saw 3D bioprinting mentioned prominently in the texts. If the stem cells are so good at self-organizing through that MT process we talked about, why do we need to 3D print them? Isn't biology doing the heavy lifting?
Biology is doing the heavy lifting, but, well, biology is inherently chaotic. It comes down to reproducibility and scale.
Okay.
When you rely solely on self-organization in a suspension dish, there is inherent variability. One organoid might spontaneously develop 50 nephrons, while the one next to it might develop 200.
That would mess up a drug test.
Exactly. One might have an overabundance of stromal tissue, the connective tissue, while another has too little. If you are doing high-throughput drug screening, you need a baseline. You need thousands of organoids that are mathematically, volumetrically, and cellularly identical. Otherwise, your data is just noise.
So 3D bioprinting imposes architectural order on the biological chaos.
That is its exact purpose. Researchers are now using highly specialized 3D bioprinters. Instead of plastic filament, these printers are loaded with what we call bioinks.
Bioinks.
Yes, these bioinks are complex hydrogels filled with billions of specific kidney progenitor cells mixed with extracellular matrix proteins to give them structural support.
It's essentially biological spackle.
A very smart spackle. The bioprinter extrudes these bioinks with pinpoint, micrometer spatial control. It lays down the cells in highly reproducible grids, or even prints them into specific anatomical shapes, ensuring that every single organoid on a 96-well plate has the exact same starting geometry and cell density.
That makes total sense. It reminds me of the Unimat platform mentioned in the literature. It seems like another way to impose perfect environmental conditions.
Yes, the Unimat platform is a perfect example of overcoming the geometry problem. It's a 3D, geometrically engineered, permeable, membrane-based platform.
How is that different from a normal dish?
Well, normally an organoid sits on a solid plastic bottom. The top gets nutrients from the fluid, but the bottom is suffocated against the plastic.
So it's uneven.
Unimat changes that. The organoids sit on a highly permeable membrane. This provides an unrestricted 360-degree supply of soluble factors, oxygen, and nutrients from all sides.
It's like hydroponics for human organs. You suspend the roots in a nutrient myth so they grow perfectly evenly.
The data supports that analogy perfectly. Organoids cultured in these permeable, dynamically flowing environments show a much more in vivo-like cell type balance. You don't get a necrotic core where cells died from lack of oxygen, and you don't get an overpopulation of connective tissue. You get scalable, highly uniform, mature organoids ready for rigorous scientific testing.
So what does this all mean? If we step back, we are effectively building microscopic, robotic life support systems to force these mini organs to grow up. We are merging plumbing, architecture, and biology.
That is the multidisciplinary reality of modern regenerative medicine. But, it raises a monumental data problem.
A data problem.
If we are using bioprinters and microfluidic chips to produce thousands of organoids, and each organoid contains hundreds of thousands of individual cells representing 20 different cell types, how do scientists actually track what's happening inside them?
Oh, wow. That's a lot of cells.
Right. If you drop a toxic drug onto that chip, how do you measure its effect on the podocytes versus the proximal tubules simultaneously without just drowning in an ocean of biological data?
Enter the map makers. This is where the sheer computing power comes in.
Exactly.
Because to truly understand these organoids, looking at them under a standard microscope is nowhere near enough. You need to know exactly what every single cell is doing, which genes it has turned on and which genes are panicking. And for that, researchers use a revolutionary technique called single-cell RNA sequencing, or cRNA-seq.
It is arguably the most powerful analytical tool developed in the last decade. To appreciate it, we need to understand how it differs from the old way of doing things, which was bulk RNA sequencing.
The analogy I always use for this is a fruit bowl.
Okay, let's hear it.
Imagine you want to understand the exact nutritional composition of a complex fruit bowl. Apples, oranges, bananas, grapes, maybe some blueberries hidden at the bottom. Bulk RNA sequencing is like throwing the entire fruit bowl into an industrial blender, hitting puree, and then tasting the resulting smoothie to guess what was in it.
That's all mixed together.
Right. You know there's fruit. You can generally taste the dominant citrus and maybe the apple, but the delicate flavor of the blueberry is completely lost in the noise.
The smoothie gives you an average. In biology, bulk sequencing gives you the average gene expression of the entire organoid.
Which isn't enough.
No, because it was bulk RNA sequencing that initially revealed the uncomfortable truth that early organoids were only maturing to the equivalent of a first trimester fetal kidney. The average gene expression was immature. But the smoothie couldn't tell scientists which specific cells were lagging behind, or if some cells were fully mature while others were completely broken.
So single-cell RNA sequencing changes everything.
Yes, it's like taking the fruit bowl and meticulously examining every single piece of fruit, weighing it, measuring its sugar content one by one.
But how do you actually do that to 100,000 microscopic cells? The engineering behind it is wild.
It relies on droplet microfluidics. First, the solid organoid is treated with special enzymes that dissolve the glue holding the cells together. The organoid is enzymatically dissociated into a liquid soup of individual free-floating cells.
You unbuild the wall back into a crowd of people.
Exactly. Then, that cell soup is pumped through microscopic channels. At a specific intersection, a stream of oil pinches off the fluid, encapsulating each individual cell inside its own tiny microscopic droplet of water.
One cell per droplet.
Yes. And crucially, injected into that droplet along with the cell is a microscopic bead covered in a unique molecular barcode.
A barcode, like scanning groceries.
Conceptually, yes. Inside that tiny oil droplet, the cell's membrane is popped open, and all of its RNA, the instructions it was currently reading, spills out. The molecular barcode attaches itself to every single piece of that cell's RNA.
That's genius.
Then you can sequence all the RNA from all the cells together. Because of the barcodes, a computer algorithm can sort through millions of genetic transcripts and trace every single piece of RNA back to the exact individual cell it originated from.
That is staggering. You literally map the exact transcriptomic state of every single cell. You know what the podocyte was doing, you know what the distal tubule cell was doing, completely independently of each other.
You map the entire cellular landscape. This mapping has allowed scientists to identify exactly which specialized cell type successfully formed. But perhaps more importantly, it allowed them to identify the stowaways, the off-target cells.
Off-target cells.
Yeah.
Like cells that wandered into the wrong neighborhood.
Precisely. Remember the cookbook. We start with pluripotent stem cells that can become anything. The biochemical recipe, the Wnt signaling, the FGF9, is designed to force them down the kidney path. But, as we said, biology is messy.
Things go wrong.
Right. If the timing is off by a few hours, or the concentration of a chemical gradient is slightly weaker on one side of the dish, some cells might wander down the wrong developmental path. Single-cell RNA sequencing revealed a shocking reality. Early organoid protocols were accidentally generating stray cartilage cells, muscle cells, and even melanocytes and rogue neural cells mixed in with the kidney tissue.
Wait, a rogue brain cell trying to fire synapses inside a kidney organoid.
Yes!
That's both amazing and terrible if you are trying to test a kidney drug.
Exactly. But because they used seqRNA-seq, they could finally see the stowaways. The smoothie approach hid them. The individual fruit approach exposed them.
And because they could identify the stowaways and track their genetic history, they could fix the recipe. Researchers developed computational classification tools like DevKidCC, which is a machine learning algorithm specifically trained to recognize human kidney organoid cell types.
A very powerful tool.
By analyzing this massive single-cell data, researchers iteratively refined their biochemical recipes. They adjusted the exact hour they added the Wnt inhibitor or tweak the concentration of FGF9, actively eliminating those off-target cell trajectories. They refined the protocol until the organoids were pure kidney tissue.
It's iterative perfection based on massive data sets. But analyzing the internal RNA is only half the battle. When you're running those high-throughput drug screens, testing thousands of drugs on thousands of organoids, you also need to see the physical changes.
Did the cyst shrink? did the tubule collapse.
Right, and you can't have a graduate student manually looking through a microscope at 100,000 organoids, sketching what they see.
No, that would take forever.
The bottleneck of human observation has been shattered by artificial intelligence and machine learning. We are seeing the integration of automated, high-throughput 3D imaging systems. These systems use advanced physics, like tomographic phase microscopy.
Let's break that down. Tomographic phase microscopy. How is that different from just taking a really good picture?
Standard microscopes often require you to dye the tissue to see the structures, and those dyes are usually toxic, which kills the organoid. Tomographic phase microscopy uses the physical properties of light. As a beam of light passes through a transparent cell, the dense parts of the cell, like the nucleus, slow the light down just a tiny fraction of a second compared to the fluid around it.
So the light actually slows down.
The microscope detects these microscopic shifts in the phase of the light and mathematically reconstructs a highly detailed 3D image of the live, living organoid without ever touching it or dyeing it.
So you have a robot arm moving a plate of 1,000 organoids,
Yeah.
flashing light through them, and generating massive 3D models in real time. And then they feed those 3D models into an AI.
Yes. Deep learning algorithms, specifically convolutional neural networks, are trained on thousands of previous images. The AI learns to recognize the specific morphological features of a healthy organoid versus a sick one. It measures the precise volume of a fluid cyst. It tracks the thickness of the tubular walls. The AI can automatically analyze thousands of 3D images overnight. It detects the earliest structural signs of cell death hours or days before a human observer could spot them.
That is just wild.
It predicts drug toxicity with a speed and mathematical accuracy that fundamentally changes the pace of discovery.
Think about this, directly applying it to you, the listener. We've talked about a lot of abstract biology today, but imagine a future, maybe just a decade from now, where you are diagnosed with a severe, potentially fatal kidney condition.
A real possibility.
Your doctor doesn't just guess which drug might work based on statistical averages. They take a simple blood draw. From those blood cells, they reverse engineer your stem cells. They generate your specific, genetically identical kidney organoids. They place them in a microfluidic chip to simulate your blood flow. And an AI runs thousands of drug simulations on your miniature kidneys overnight.
It's incredible to think about.
By morning, the AI has found the exact medication at the exact dosage that will cure you without causing any side effects to your specific biology.
It is the absolute pinnacle of personalized precision medicine. We are moving from reactive medicine to predictive simulated medicine. And the thing is, it is not science fiction. The foundational architecture for that future is being built right now, piece by piece, in laboratories across the world.
Which brings us to the ultimate goal. The holy grail of all this research.
Tell me one.
Disease modeling, AI drug screening, fixing genetic mutations in a dish. These are incredible immediate applications, but they are stepping stones.
They are. The long-term vision, the true promise of regenerative medicine, is the creation of fully transplantable, bioengineered human kidneys. We want to be able to build a macroscopic, permanent replacement organ from scratch to clear that transplant waiting list forever.
We've talked about how amazing these organoids are, but how close are we really to a transplantable organ? What's standing in the way?
We are still overcoming significant engineering challenges, mostly related to scale and plumbing. While microfluidic chips help with vascularization on a microscale, getting tiny capillaries to form plumbing those tiny vessels into a major macroscopic artery and vein that can be surgically sutured into a human patient and withstand the immense pressure of the human heart. That is a massive bioengineering hurdle.
Right, if the plumbing isn't perfect, the blood pressure would just blow the microscopic vessels apart.
Furthermore, organoids currently lack a macroscopic urinary drainage system. They filter fluid, but they don't have a singular ureter to connect to a human bladder. The fluid they filter just pools in the dish.
Oh, right. So we need a main drain pipe.
Exactly. Figuring out how to coax millions of tiny distal tubules to all connect to one central drainage pipe is the next great developmental puzzle.
But assuming we solve the plumbing, and given the pace of bioengineering, it seems like a matter of when, not if. The sources suggest a very clear path forward regarding the immune system. Organ rejection is the biggest hurdle in traditional organ transplants. A patient gets a donor kidney, but their body views it as a foreign invader and tries to destroy it.
It's a lifelong battle. Patients on transplant lists must hope for a match in blood types and HLA human leukocyte antigen
What
profiles.
are HLAs exactly?
HLA are the proteins on the outside of your cells that act as a biological ID badge. If the donor kidney has the wrong ID badge, your immune cells attack. Even with a good match, transplant recipients must take heavy, debilitating immunosuppressant drugs for the rest of their lives, leaving them vulnerable to infections and cancers.
But with organoids, we can use that CRISPR gene editing technology we talked about earlier to alter those ID badges.
We can create what are called HLA-edited or hypoimmunogenic stem cell lines. By using CRISPR to knock out the specific genes that express those immune recognition proteins on the surface of the cell, we can essentially render the cells invisible to the human immune system.
They become biological stealth bombers. They fly completely under the immune system's radar.
Essentially, yes, this is a monumental concept. It means we wouldn't necessarily have to grow a custom kidney for every single patient from their own blood cells, which takes months. By using these invisible hypoimmunogenic stem cells, we could create universal organ building blocks.
Universal.
Yes, we could biomanufacture massive vascularized kidney tissues in advance, knowing that any human body would accept them without requiring a lifetime of toxic immunosuppressants.
We've covered an incredible amount of ground today. We started with the basic understanding of our biological limits. We watched a reprogrammed stem cell get coaxed through the biochemical origami of directed differentiation.
It's quite a journey.
We saw it self-organize into a complex, functional nephron. We watched scientists use CRISPR to intentionally break it to model deadly diseases like ADPKD. We hooked it up to microfluidic chips to simulate the friction of blood flow, forcing it to mature. And we mapped its entire existence using droplet microfluidics, single-cell RNA sequencing, and artificial intelligence.
It is a testament to the convergence of biology, mechanical engineering, and computer science. No single discipline could have achieved this alone.
It really is. And I want to leave you with a final thought to mull over. We started this deep dive talking about the fatal flaw of the human kidney, how its regenerative capacity has a hard stop before you are even born. We are all born with a biological warranty that eventually, inevitably, runs out. But if we can master the art of coaxing stem cells to build complex, immune-compatible replacement filters for our bodies, we aren't just solving chronic kidney disease.
No, we're doing much more.
We are fundamentally redefining the human warranty period. If your most complex, irreplaceable internal parts become easily replaceable, what does that mean for the natural limits of human lifespan?
It is a profound philosophical and biological question, and one that the next generation of science will undoubtedly have to answer.
Thanks for diving deep with us. Keep questioning, keep learning, and we'll see you next time.